Good afternoon. I'm very glad to be here to present to you our latest work. But first, since my presentation will regard the movie, I would like to start with a sound that probably most of you will recognize. And since last year at OHBM in Singapore, I presented the first part of this study that is now a preprint. So if you're curious, you can find it by Googling emotionotopy. And the focus of our study is on emotions. And emotions are at the same time so fundamental and still so confusing and difficult to properly investigate, since they are very complex descriptions of inner events that involve several components and that also have a specific time course. So emotions have onset and offset, and they're not related to just a point in time. So to properly study how emotions are represented in the brain, it is important to take into account both their complexity and their dynamics. And one way to do so could be through the use of functional connectivity that takes into account the interaction between different brain regions, and through the study of functional connectivity dynamics, which is a metric that instead consider also the dynamics of these interactions. And previous studies in the domain of the emotions actually focused on the peak of the emotional percept, so without acknowledging the following cascade of events that are what at the end characterize the emotional experience. Some of them employed metrics of connectivity in time to try to overcome this issue, but they only investigated a subset of affective states, since it is indeed very difficult to consider both the complexity and the dynamics of emotions. So what we aimed to do in this study was to investigate which brain regions are associated to subjective reports of emotional experience, and then to measure to what extent the dynamic interplay of these brain regions is related to moment-by-moment changes in the emotional experience. And to overcome the limitations of previous studies, we used 2 hours of brain activity recorded during movie watching, thanks to a publicly available dataset that had subjects watch the movie Forrest Gump in the fMRI scanner. So what we did was thoroughly pre-process the fMRI data. We averaged single subject time series, and we applied a temporal smoothing with a 10 seconds moving window. And from the obtained time series, we also regressed out the low-level physical properties of the stimulus. So we regressed out the RMS for the audio signal and the low and high spatial frequencies. What we did then was to ask a group of subjects to watch the exact same movie employed in the fMRI experiment, and to give a continuous ratings of the perceived intensity of 6 basic emotions, which were happiness, surprise, fear, sadness, anger, and disgust. And subjects were free to choose as many emotions as they thought it was appropriate for their experience. So we then, first of all, we checked the goodness of our behavioral experiment. So we measured the concordance across subjects in their ratings, and we found an average correlation of 0.4 over 3,600 time points. And then we also checked back in the movie which were the points associated to the peak of affective states. And for example, we found that the peak of sadness felt by our subjects happened during the scene in which Booba dies, so it made sense. So what we did then, we pre-processed the behavioral data following the same method of the fMRI ones, and we applied a principal component analysis on the group average time series, which highlighted the existence of 3 emotion dimensions that together explain more than the 80% of the variance, and that we interpreted as describing the polarity, complexity, and intensity of the experience. So polarity, we have positive versus negative emotions, so similar to valence. Intensity, we have the same type of load of the 6 emotions, so resembling arousal. And complexity, we interpreted in this way, since we have on one side happiness and sadness together, so denoting a state of inner conflict and ambivalence, something more cognitively mediated, against more instinctual states such as fear. So to test then the association between our emotion ratings and brain activity, we used our 6 components as our full model to encode in the brain activity using a voxel-wise encoding, and obtained the map of the fitting. And we then, with a phase shuffling method, we generated 10,000 maps of this fitting that constituted our null distribution, on which we computed the p-value, then corrected for multiple comparisons. So these are the regions found to be significantly associated to the emotion rating model, so regions mainly in the right hemisphere and already found in the previous literature. And then, starting from this map, We then extracted for each pair of voxels their time series. We windowed them using a 50 time points window, and there computed the functional connectivity dynamic matrix, so their association in time. So we obtained a final time series reporting this matrix that we then related to the time series of polarity and the other dimensions, building a correlation matrix reporting this association. To build a graph from this matrix, we then generated 10,000 null models of each motion dimension time series using a phase shuffling method, so that we had for each cell a null distribution reporting the number, the correlation values. So then we obtained our graph by thresholding the original matrix using the arbitrary value of p0.05, and this gave us two important values. First of all, we had an overall number representing how dense our graph is, and then locally, so within each parcel of the matrix, we have the number of associations, so how much the coupling between two regions is associated to polarity, complexity, and intensity, and this holds for the values that are off the diagonal, while on the diagonal we have voxels pertaining to the same region. And then, so we had the problem to test the statistical significance of this association, and to do so, we use the overall number of occurrences obtained from the thresholded matrix to create null matrices having the same overall density of the real one, and then we counted for each section the number of occurrences under the null hypothesis of significant cells being randomly distributed across the matrix, and these numbers populated the null distribution against which we tested the real number of occurrences, and we then computed the actual p-value using tail approximation and corrected for multiple comparisons. So what we found was that the functional connectivity dynamics of the right precentral sulcus and the right occipitotemporal sulcus was significantly modulated by changes in the polarity dimension. And it could be that since the OTS is actually involved in the processing of phases, the connectivity between these two regions might be useful for decoding the positivity or negativity of facial expressions. While we found that the functional connectivity dynamics of the right precentral sulcus and the medial superior frontal gyrus was significantly modulated by changes in the intensity, and the medial superior frontal gyrus is involved in the perception of fearful body expression and painful facial expression, so these results seem to be in line with the fact that the changes in the intensity of emotions modulate the connectivity between these two regions. And lastly, we found that the intrinsic connectivity of the right precentral sulcus was significantly modulated by changes in the complexity dimension, so a dimension reporting more cognitively mediated states versus more instinctual. And also, when we looked at the relative contribution of each emotion dimension to the Ball signal, we found a peculiar organization of the precentral sulcus where we have separate yet adjoining subregions coding for distinct emotion dimensions. So we have a posterior part related to intensity, a central to complexity, and an anterior one to polarity. Furthermore, the precentral sulcus seems to be involved in the perception of dynamic facial expressions and also when watching emotionally charged movies. So it seems this region seems to have a central role in this regard. So to conclude, we found that changes in the polarity, complexity, and intensity of the emotional experience is associated to connectivity dynamics of the right precentral sulcus, which is a region that also shows a peculiar spatial arrangement in association with our emotion dimensions. And with this, I would like to thank the Study Forest project, the MOMI lab at IMT in Lucca, and my advisor, and you for your attention.